Hi, welcome to Art of Academia, a weekly podcast featuring interviews with leading scientists and insider takes on life in academia, hosted by Komal and Madan, researchers from Cancer Science Institute, Singapore.
Hello, what's up?
What is up? Well, we're back recording. That's good.
Yes.
It's been a while, I guess, which we say every time. But what has happened since last week? I don't know if this was already the case, but I got my defense date.
Yes, congratulations.
Yes. So
Something to celebrate, I guess.
Something to celebrate, yeah. But the main event is coming soon. I have to
in 2 weeks'
defend
time.
In 2 weeks' time.
No, actually, we can talk about that as well. How does that feel? So you mentioned that you're going to take a 2 weeks preparation time. What's that going to look like?
To prepare for the defense. Well, I don't know. I haven't started yet.
Okay.
Yeah, so hopefully don't release this until my defense is over, just to lower the chance that somebody hears it. But I don't know. It's supposed to be like this grand thing, right? But I'm not feeling it. Maybe I think on some level, I am suppressing it probably, right? Because I'm not sure why. But it's just not hitting me.
Yeah.
But
I feel like in a way it's maybe better to not be overwhelmed by it.
Yeah.
Than not feeling anything about it.
The funny thing was, Dennis, my mentor, the other day, he walked into me into the lobby and he was like, "Oh, how are your, something like, oh, your cortisol levels must be very high or something." And I was just like, what do you mean? Like, I'm doing fine. And he's like, "Your defense." And I was like, "Oh, right." And I was like, "No, not yet. I'm okay as of now." And he just like gave me this weird smile and he walked off, which I thought was quite funny. Yeah, I don't know. How was that for you? Did that feel momentous in the building up to it?
Yeah, I think for me, the writing thesis part was sort of a bigger challenge.
Yeah.
Because in general, of course, it just took so long, and then there was just so much work that was put into it. So just submitting that felt like a major relief. And then for me, for a long time, the date of defense was not decided, and then it got suddenly decided, and then I just had like a week or two, something like that, before knowing the date and then actual defense. And it was also online, so in that sense, I guess a bit different vibes, because there's no audience. It's just like you in a meeting on a Sunday afternoon.
Yeah.
In the same room.
Oh, it was here. You took it from here. Yeah.
Yeah, it was in the same room. Yes. I think I was doing okay until I did like a mock with my lab. And that was a little scary, because in general I feel like some of the take-home messages were not as strong as of then. But that is the case with like a lot of theses, I think. And eventually the defense itself was pretty chill. There were obviously some questions that I just could not answer, but in general, for defense for us, there was no presentation.
Oh, right. You don't have a presentation.
Yeah. So they just asked me to do like a 10-minute recap kind of thing. I don't know if they even listened to it. So it was just like a summary of the entire thesis in 10 minutes. A presentation online, like I just shared it. Spoke for 10 minutes. And then after that, it was like an hour or two of just questions. So they would really read the thesis like a hawk. And then they would go through it, page this, line this. What do you know from this reference? Or like...
Wow.
Yeah. Sometimes that could be like that. Some of the examiners are really very absolute pain in the ass. But so they would be like going through it chronologically and then be like, oh, this plot, explain it, or like what controls are missing, or like anything more philosophical about this result. What does it mean? What does it mean in the context of literature? What about BRCA2 heterozygosity in other systems, models, etc., etc. So then eventually it was just a bunch of questions through the thesis itself. And then by the end of it, I think did they kick me out, or they, because it was a Zoom meeting.
Zoom meeting, yeah.
Yeah, or they just like, I'm not sure how they discussed. Maybe I can't remember anymore. They kicked me out somehow, and then they had a quick discussion, and then came back and they're like, "Oh, congratulations, you have it." And
Dr.
then,
Gupta.
Yeah, I did not feel much actually at that point. I was obviously like very happy, but also I was just alone in this room. So it was also a bit strange. I think it's nicer to celebrate and then just get that feeling a bit more. But yeah, that was it. I did not really feel... I think before the actual thesis defense day, like in the morning, I was anxious. I meditated and all, and then I was like, okay, let's go, let's do it.
Yeah.
But that was it, yes.
Yeah, I think it will kick in for me this Sunday. I hope it does. It's tomorrow. 'Cause
Yeah.
that's time.
But it's also like, what do you even prepare? Like, in a way, this has been your work.
Yeah.
All you could do is just read more literature and for me, it was like just reading my own thesis and just knowing everything that I'm putting in there.
Dude, I said so many things. I better go back and read what
Yeah.
I wrote. But yeah, I think maybe in the preparatory phase I don't have much insights to give. So maybe we can come back to this after I'm done.
After the preparatory phase has been gone through.
Yes, yes.
Yeah, cool stuff.
What about you?
What
have
about
you
me?
been up to?
Yes, this week was actually very busy. So, as I mentioned in the last podcast, I was attending this conference, FCS, Frontiers in Cancer Science. And for I did not prepare my poster for the longest time. And I just crammed that in 2 days. So, Monday, Tuesday was just like a bit sleep deprived, just trying to get everything together. But that really helped me because now, obviously, all the scattered things that I had in the last few months are in one place now, in some sense. And then there's a narrative to it. So, that definitely helped me. The conference itself, okay. Some nice talks. Like, I attended day one, all the talks there. 2 talks were quite nice. Again, completely depend on the speaker. This one lady and this guy, they just presented well. They just put everything in a context that is interesting to everyone. They were enthusiastic about their work. So that was quite cool. And then day 2 was my poster. So I think that is the most enjoyable part for the conference because it was also a lot of catch up because now I'm not a part of CSI where I was working before. Just meeting all of those people and they would all drop by my poster and in general other people as well who would just now come and ask questions. So in terms of the work itself, it's not very paradigm shifting or making new hypothesis kind of research. It's more clinical and it is more a bit descriptive as of now.
You're talking about your work?
My own work.
Okay.
Yeah. So in that sense, there were not a lot of questions in the way how does this work or what do you think about what hypothesis is there kind of thing. It's more data driven as of now. But I definitely enjoyed it. And this is also something I was talking about earlier to you about sharing your work early.
Yes.
Because this has only been 5-6 months that I've been working on this, but just sharing it gives you, of course, some feedback from the public. Generally, how important this work seems, how interesting this work seems, how can you improve on it? Because I did get some feedback from good stat people and people in clinical field who could come and just tell me things from their experience. Because eventually, while you're working on your thing, you get in that bubble.
Yeah.
You're talking about the same thing again and again with the same people who have the same idea in their head, the same literature in their head. So that in that sense, I definitely got some value.
And if you think about it, I guess that is the main point of conferences, right? It's just to talk to people.
Yeah.
This whole giving talks. I don't know, when you're new to conferences as a young PhD student, maybe even RA, because this time in our lab, none of the PhD students went actually. It was just the RAs, right?
Yeah.
And they always approach it from what can I learn from this talk? Which is true. I guess partly that's why talks are for.
Yeah.
But if you look at what the PIs are going there for, it's never so much of what new biology did I learn, right? It's more about who's doing interesting stuff, who can I connect with.
Absolutely.
A lot of them they are presenting. So it's a lot about what feedback can they get for their work.
Yeah. Because eventually their talks also are mostly bioarchived papers at that point. It's not something, very rare people are showing very unpublished work.
Yeah.
Even if they are, it's just exactly as you mentioned. It's seeing, they did do this, how can we connect? How can we now think about or learn about this cool thing?
Yeah, and that's why also I guess it's not tailored it's not a good learning experience because usually people are trying to fit in everything they're doing, which again is not a great thing to do to the speaker, to the listeners. But I kind of get where they're coming from, right? They just want to expose as much of their work as possible, hoping that somebody finds some bit interesting.
Yeah.
Right?
Yeah.
So,
but that is also an absolute art, I feel, to be able to now have all of this research, keep it of course you have to show data, of course you have to show insights, but keep it in a way that it's easy to comprehend.
Yeah.
As opposed to just flashing data after data, which was happening. And especially now these days, the day 2 talks shouldn't be bashing too much, but there's so much single cell stuff. And that always tends to be very data heavy, information heavy. So it's just bunch of plots showing different cell populations and whoever is in the field, of course, appreciates it more. I am not one of them. But a lot of times it feels to me like, oh, tumor microenvironment. Every talk is the same.
Yeah.
Just like a different disease or a different population of cells, and then tell me what it means.
Yeah.
I'm here to listen to you for like half an hour. Give me one line line summary of
What you're doing.
what can I what can I understand from all of this cool stuff that you're doing.
Yeah, I think it's interesting the way conferences are held. I feel like my take on it has changed over time, but I still don't feel like I get it. Like, I don't think I'm a good conference participant, So yeah, maybe we'll see. Maybe this is something you figure out as you go along the way.
Yeah.
Cool. What
In
else?
terms of the beta part also, I feel like this
The
is
what part?
being in constant
Oh,
beta.
okay.
I should have actually explained this more. This is from
Yeah, I know
the
what
essay.
that's from, but I'm
But these people
surprised
don't
that you
know.
threw that in like that.
I think this is a different topic, but in my previous lab, I used to present less frequently, like every once in 2 months or even sometimes longer. You're just doing things, and obviously experiments take long as well, so maybe that time is enough. But now I'm presenting much more frequently. And initially, I remember I used to tell you or like other people who have been presenting more frequently, like, oh my god, that seems so painful. But I feel this is actually really helping me now just shape things very quickly. It's also like constant feedbacks and constant things to look forward to and work faster, because I talked about this in this meeting, but within 2 weeks' time, I'm going to meet again. So I want to really, get that done and get it going and then talk about it eventually. So I think that's also helping me in some sense.
Yeah, that's good. I think definitely, I don't know if all labs run it this way, but I do feel on average computational labs meet more frequently, right? And that makes sense.
Yeah.
Okay, cool. All right, let's move on.
Yeah.
Okay, what do we want to talk about today?
We can talk about the Reddit page that you've created.
Yeah. Okay, yeah, so I guess I actually remember this. This was in a very old podcast that we did, I think. And maybe there were scenes, right?
Okay.
Where the idea was to just get couple of like-minded people. At that time, I was thinking exclusively bioinformatics.
Yeah.
But to be honest, that was never like a core element. The idea was more that you gather some people who have like similar interests, put them together, and then just have them do something together, right? Ideally, that's like work on one project, but it doesn't even have to be that. It could just be like a community kind of a thing to get the conversation going.
Yeah.
So at that time, we were thinking about places like that. I even forget what it's called, but there was this like writers cafe, right? There was this cafe where all the greats would meet, and they would just throw some ideas off of each other, and how they all felt that was crucial to their element. Basically, the whole scenes episode. So that's what I was talking about recreating. And I guess we tried that. I did like a Discord server once.
Oh yeah, what happened to that actually?
People, yeah, but
was I ever? I don't even know if I was added.
I don't know if I added you, but I did I think I added like whoever was on Discord, so maybe Andy and all. But no, the problem was I didn't know what to do with the platform. So Discord is normally meant as a community oriented tool, but I didn't have a good sense of what we would be doing there. So at that time I was thinking we would do we could have some work together sessions. So at some point you just say that I'm going to be on the server. I'm just doing my own thing with my camera on, and people can just join in, right?
Like just working together remotely. You can have a chat.
Exactly. There was that, and then
just hang out.
We floated around this idea of you would have a channel where every day you just put out your main task for the day or something. And then others would check up on you to have some sort of accountability. There was a few ideas, but somehow the execution it did not really land well, because I felt like that requires participation. Obviously, right? You can't do that alone. You need people to do that with you, and if everybody is not excited, it doesn't work.
Yeah.
So that kind of
then it's not the scenes idea.
Yeah, exactly.
To begin with,
right? Yeah, you need everybody to be equally interested, I guess. But then randomly recently, a couple of weeks ago, I was talking about sort of the same complaint, and I don't know how I even had this thought, but it was just a random work day, right? And I was like, it would be so nice if we could share whatever we are learning every day on some platform, right? And then you can also get to read other people sharing what they are learning.
Yeah.
And that does two things. One, I guess you can learn more from whatever other people are sharing. But more so, it's also just like getting small wins along the way, I guess, to like push you forward. You feel like you're like making progress.
Yes.
Blah blah blah.
Yes.
Just learning in public, basically.
Correct.
So I just said that out loud on like this group chat that we have, me and two friends, and then they were very enthusiastic about it. They were like, "Oh yeah, let's do it." And then I guess that enthusiasm caught on, and that eventually translated into making a subreddit, a Reddit community. I don't even know what you call it. I guess just a subreddit.
Yeah. What is it called?
Yeah, so it is called Spill the Tea, but TAE, like TAE buffer. This was Kaisa's pun, so let me put that
I like
out
it.
there. Yeah, big on puns. So I'll just read the sort of welcome message we have, right?
Yeah.
Which is sort of the motto, I guess. So this is a space to share where you learn from your research every day, whether it's a fun hack, a late realization, or a pro tip learned from a fuck up. We believe that even the smallest discoveries are valuable and worth sharing.
Yeah.
So that's about it, right?
Yeah.
It's like whatever small stuff you learn along the way, something fun, some mistake you did, whatever, just put that out there. Nothing is like too lame or like too small. And hopefully when enough people do this gains some mass. That's all. Very simple.
Yeah.
Started out like that. And how many? We are like 7 members right
Nice.
now.
Let's go.
And I would say we're doing well.
Yeah.
We have like a decent number of posts, some good ideas. So today we thought we'd just go through some of these.
Some
posts.
of them, yes.
Okay, you can choose. You start first.
Maybe do you want to do the first one?
Just the very
because
first
it's
one.
just
Okay.
you. Yeah.
Okay, I guess that's actually a good start because that was something very silly. Right? It was just I was just doing some experiments which involved taking like a drug and adding that to my cell culture. Right. I had some and drugs usually they are solubilized in this compound called DMSO, which is good at solubilizing everything. So I had just done that, right? I received my drug, I took some DMSO, I mixed it up, and I kept it on ice, which is a normal thing to do because you want to preserve the integrity of compounds. And then when I went back, I did my TC, and when I came back just like a few minutes later to take my DMSO now with my drug to add it, and it was in a like non-see-through tube, opaque tube, right? So I couldn't see inside.
Yeah.
But I knew it was there. So I just reached in with my pipette and I pipetted and nothing's coming out. And then I was like, wait, what happened? Did I not make it? Or something happened? I thought. And then that was, first of all, a good 5 minutes of confusion. Like, I'm pretty sure I made it. Why is it not
Did
here?
I really not make it?
Yeah. And then I realized it's actually frozen. Frozen solid, like ice.
Yeah.
Just from keeping that on ice, right? Which normally doesn't happen.
Yeah.
Then I went to look it up and I realized DMSO has a super low freezing point, a super high
freezing point, yeah.
Right? Which is like at 20 degrees.
Oh.
Which is ridiculous.
Right, that's the title of your thing. Yeah.
I was like, why is this liquid freezing at 20 degrees?
Yeah.
There's no business doing that. But good to know, I guess. So then I thought it again and then I added it.
20 degrees is really high.
Yeah. That's what I mean. I literally put it on ice for like a minute or two.
Yeah, like on ice I understand, on ice, because I have done it before. And I know on ice it freezes. But 20 degrees is actually very surprising.
Yeah, I think
sometimes the lab temperature is just 20 degrees.
Yeah. I think the actual one is like 18 point something. I just rounded it.
Sure.
But yeah, that is pretty high.
Yes, that was the first post that sort of started off this page, I guess. And then there are a bunch of people talking about just general lab. Plastic waste in the lab. This is again you. Do you want to just do it?
Do one of yours, then I'll do
Okay, can. Yeah. So I think I joined the page a few days later, and then for a while I was actually just like thinking on what to do, and then I was just jotting down some points, and then and one night I was just like, okay, let's go, and I went crazy and I just like posted a bunch of them, and I don't know if I posted after that at all. I have, I think. I'm not sure. Okay, so one of the posts that I wrote was about this thing that is happening in the lab, just this project that's ongoing. So during one lab meeting, this girl was presenting about her organoid project. What they do is they get some tissues from patients and then they're trying to make these organoid systems in the lab. So then eventually they can test drugs on them, etc., etc. It's just valuable biobank kind of thing that they want to make. But every time they would collect these patient samples, which are supposed to be having a mutation in a gene EGFR, they would grow, and then eventually, after a few weeks or so, they sequence it to test the mutation, and it's gone. And that was what she was worried about and like perplexed about what to do about it. And then she was just sharing the data, and then eventually the PI was just like, we have been working on this for 10 years. We are trying to generate this organoid system with EGFR mutation for the last 10 years, and just we are not successful. So I was just like, damn, this is like, such a long time to be working on something which is impressive but also scary in the same in a way. So then we were also like talking about what could be going on.
Yeah,
what could
why does it disappear?
Yeah, so we thought about a few things, and I think now actually, I think they actually have some updates. I'm not sure exactly what, but I think they are towards figuring it out in some sense. So I think maybe I should update it once I know the solution and if it actually works out. I think it's something to do with the actual sequencing at the patient level. Because I think recently they just got the patient tissue, and the clinicians say it has EGFR mutation, and then instead of putting it in the organoids, they directly sequenced it with whatever their system. And I think they did not see an EGFR mutation to start with.
I see.
Which was a concern that I was raising also. But I was like, it can't be true. They have tried it so many times. It can't be always that it actually doesn't have the EGFR mutation. So I guess they will hopefully figure it out.
Yeah. And you don't even have to see, can't you just do like PCR?
It is Sanger sequence. Yeah, it is just targeted
okay.
for that gene. It's not a whole genome exome sequencing anyways.
Yeah, that should be very easy to
No,
test,
the
right?
problem is they don't do it because the tissue is precious. So they don't want to split too much because they want to use all of it for the organoid establishment. Because volume matters, the size of the tissue matters and stuff like that. And in general, I feel like organoid development is just so hard. Like the smallest nutrient component is missing, and then they're like, no, I'm not gonna grow.
Yeah,
I'm done with life.
Makes sense, right? This is also why something like IVF in comparison looks so lame, right? Like, look at these guys.
Oh.
Bro, they're taking a piece of your tumor and turning that into a whole organ, man.
Yeah.
That's way more interesting than one egg and one sperm fusing
Into a human.
No, that all happens in the body. Only the fusing happens in the
Fair
test
enough,
tube.
yeah.
Okay, what else do I like? Well, okay, I'll do this one because I did find that quite cute.
Yeah.
So, context, I'm working with bacteria now. Okay, new arc. And for one experiment, I needed to isolate genomic DNA. Simple, basic procedure in the lab. But then now I'm like, wait, how do I actually do this for bacteria? Because they have cell wall, right? I can't just use our normal genomic DNA extraction kits because they don't deal with it because mammalian cells don't have a cell wall. So that's not a problem. So now I'm like, oh, what do I actually do? And then in my head I was like, wait, miniprep kit? Then I remembered, we just do that for plasmids, right? The whole genomic DNA is always lost. Okay, I'll come back to this. Eventually, I did figure it out. It's actually not that complicated. You can just use some enzyme, either proteinase K or lysozyme, that gets rid of the cell wall. And then you process that normally. That works.
I see.
But I was very smart about it. There's actually a kit that does it. Then I emailed Thermo saying, oh, will you give me a free sample? And like idiots, they gave me a free sample.
Oh, nice.
I did my experiment.
Oh.
Done. We will never buy that kit. Anyway, coming back to the miniprep question, right? So then I was like, okay, maybe I should give some of the background as to otherwise why it's not interesting, right? So, okay, miniprep kits, or actually any of these kits, right? So previously, whenever you wanted to extract DNA from an organism, you did something called phenylchloroform extraction. And this was, I still don't know the exact chemistry behind it, but there is like a...
It's something like dissolves like.
Yeah, basically, right? So you get like organic components in one part of the solution, and then in another phase you get all the aqueous stuff.
Yeah.
And that way it separates things out, and you can get exactly
depending on the charge of the DNA and
and it works well, but it's a bit tedious. So then the sort of big innovation in this domain are like silica membranes, which you can make to be specific to nucleic acid. So these are just membranes that will bind your DNA, for example, and everything else you can wash away. And that makes it super convenient. Right. It's very routine. But now the question is, so, okay, coming back to miniprep. So what is miniprep? Well, most people normally use bacteria to. We talked about plasmids before, right? So again, quick recap. Plasmids are just like bits of circular DNA that's separate from your genomic DNA. And in the modern lab environment, we use these as like sort of artificial vectors to introduce some gene that we want. So let's say I want to test the function of some gene X. I'll put that in a plasmid and put that into the bacteria, and that way it can propagate it. It'll make more copies for me, and I can extract it later, blah, blah, blah. But now there is the problem of how do you get only the plasmid DNA but not the genomic DNA, right? Because they are both nucleic acids. So if you put that in these membranes, both of them will bind. So you can't really separate them. But you don't actually want genomic DNA.
You only want these circular plasmids.
So that got me thinking, and I was like, wait, I do mini prep every single day. How do I not know this? But turns out the answer is very simple. You still do the same process, but in the beginning you had an alkaline hydrolysis step. So what that does is it basically denatures all the DNA in your sample. But because plasmid DNA is kind of smaller, when you neutralize this pH of this alkaline lysis, the plasmid re-anneals. Whereas the bigger genomic DNA stays denatured. So that's the step where you spin it down and you get the white precipitate. That's all the denatured DNA that is stuck.
But cannot like a smaller chunk of the denatured genomic DNA also circularize back?
Yeah, but normally it would not be broken, right? You have the genomic DNA that's intact. So then you just put that back on a silica membrane and then you get rid of everything else. And
only your plasmids come out. Nice.
Crazy. It's a simple biochemical innovation, but I don't know. I was like, whoa, that's cool.
Like, even I did not know. I only read in your thing and I understood it right now. Cool stuff. Some computational stuff as well. Actually, this is also something I was thinking about. Like, I, of course, have been doing more computational research these days, and then there are some newer tools that I work with, or even some basic things like, say, DE-seq, which is to analyze RNA, sequencing data stuff. And I think if, I should put in the effort of writing a little tutorial or like, anything that's interesting as a post here. Given the audience, maybe I shouldn't think of that as much and just put it out.
Yeah, this is like a temporary thing, right? I imagine more people coming in the future. They might
find it useful. You also talk about how you hate PDFs.
Yeah, but you do one of yours.
Yes, and then we can come back to it. Yeah. Let's see. Maybe I can do this one. So as I mentioned, like I do more computational research now, and then because of that, there's always now a lot of data. It's just a lot of sequencing stuff that you play with, and you can bring out any kind of hypothesis. You can bring out any type of interesting insights from this huge data. And now I'm always struggling with if I want to go further into any of these details. There's like 10 things that have come out from this one analysis. I also do like multiple types of analysis, and then how many of these I really want to pull further on. So that's why I wrote this thing called this post, which was titled "To Validate or Not to Validate." What did I even say? Yeah, I think I basically just mentioned a few of the findings from my research, and just put it out there for people to sort of think on how they decide on what to pick. Because even in wet lab research, for that matter, several times you get some data, you just get some results, and then depending on the literature, depending on what's interesting, then you see a future direction in terms of what now from this result you want to follow on. So I was thinking about factors like interestingness, importance, just feasibility, I guess also, but in general, how do you sort of now decide from all these 10 threads? And also novelty. Novelty was also one of the factors, which is something sometimes a bit hard to know, because, for example, some result is seems very intuitive. I get a result which seems very intuitive. This should have been done, but it is not done in the literature. So in that case, I'm always thinking, have people tried it and failed? Or is it just like people were busy doing something else and have never looked at this very simple open question? And then I think I did get like some responses from people who were just talking about how interestingness is the most valuable, not valuable, but like most appealing factor for them to sort of
some people is
push.
me.
It is you, and also Alison, I guess, in some sense, which I do agree with, but it's just like a constant discussion in my head
Yeah.
these days.
Yeah, I'm sure maybe somebody has given this some thought and like written about it, right? In fact, that episode that we did, problem choice, was a little bit like this. Maybe
Yeah.
not exactly at this stage of your research. Like this, you're saying more is like a continual thing.
Yeah.
whereas that
Problem choice was
in the
more about like
beginning.
when you're just starting something out and then how do you choose something.
Yeah, but some elements are shareable, I guess. The reason I say just interesting, like prioritize interestingness, because maybe you already like what you consider interesting already factors in a lot of these other things of is that important?
Novelty also, I guess, because I don't think very super known things would be very exciting.
Probably not.
Because you're reading and you know what are the gaps and you know what is something that's, nobody knows.
Yeah.
And that is exciting as well in some sense.
And I guess the biggest reason to say it like that is I feel like whatever theoretical framework you might come up with as to, okay, you have all these different metrics and then you gauge your idea against these metrics and then you decide based on that. I feel like your accuracy is actually might be very low. That's my feeling, gut feeling, that no matter what analysis you might do, I find it very hard to believe that you are very true in your assessment. So that's the main reason of just saying that follow what's interesting.
Yeah.
It's almost like the analysis. What it's doing is like in my head, okay, I might be wrong about this, of course. This is like purely opinion, not fact based. But what I feel like is happening is all this analysis is doing is like giving you some comfort of, okay, I have thought about this. This is the right decision, and then now let's pursue it. As opposed to you chose without doing the whole thing.
Yeah.
In my head, they are both actually very similar scenarios. Like you're just as likely to be wrong about something.
Right.
But in the first one, you just saved yourself all this trouble of
to start
doing
with.
this analysis and you just got started. But yeah, maybe I'm wrong. We'll see, I guess.
And even for completed projects, I guess now, after looking back, you're like, oh yeah, of course I had this hypothesis all along. I knew it, I believed in it, and then obviously we proved it and stuff like that.
Yeah.
There's a lot of serendipity and a lot of movement constantly.
And now I'm thinking back to this podcast. I think this was a writer, some guy who's a very well-known writer on, I think, Sam Harris's podcast. I forgot his name. He is talking about frameworks for writing. So which is kind of similar to what we talked about. So it's like whenever he gets an idea of writing something, and then I guess how he goes about writing the piece.
Yeah.
Right. He talks about how writers, himself included, are always obsessed with like finding a system of like, okay, these are all the things I have to consider.
Yeah.
I have to do it in this way, and if it's not fitting these criteria, it's not good. I should work on something else. And it's very tempting because it does give you some assurance of like, okay, don't worry, you're doing the right thing.
Yeah.
You're on the right track, right? So everybody has this temptation to look for it, but he says that it kind of in the end doesn't matter because you end up doing what you are doing anyhow.
Yeah.
And yeah, that is the main
Fair enough.
takeaway, I guess.
Yeah, I guess it does make sense to me. Because it's also like you start off something and it actually gets interesting when you start seeing weird results. It's, you're doing it and then you of course made something in your head, and if you keep on seeing it, it's in a way boring. And you're just like, okay, I said A to B to C and yes, that is true. Yes, I did all those experiments and let's go. It's there, it's a paper. But it's actually more exciting when you started off in some way and now you're seeing interesting results, and then you play along with it.
Yeah.
In a way. And then you're just like, okay, if this is true, then what else is true? And yeah, we'll play along with the way there. I guess that's the summary.
Okay, what else? This one. Well, do I really want to do it? Okay, I guess this one is more of a rant actually, and a little bit of an advice. My personal opinion, which is that PDFs are horrible for reading papers. And this is, I feel like, holding back our field.
Of course.
Seriously.
That is why major discoveries are not being made.
I swear to God, it is so annoying. I can't be the only one, right? Like, they are just so horribly designed. Like, have you ever come across some paper and you're like trying to look at a figure, right? So the figure is on one page. The figure legend
is
for
on
some
an
reason
absolutely different
is on
page.
another page,
Yes.
and the text associated with the figure some other page. So to read one result, I have to
acrobats.
do literal acrobatics, jumping back and forth. I am like, who designed this? Okay, why is this the primary way we communicate our findings? Yeah, so that sucks, right? Okay, can we not do that? And what's the alternative? Well, HTML, which is, I think, a pretty good solution. It's not perfect, but it's just way better. It's so much superior, right?
Yeah.
I feel like most
But
journals...
HTML, you mean just the links there instead of downloading a PDF and reading through
Yeah,
it?
so that's one thing. I feel like most modern journals do a decent job of formatting their HTML interface, right? Usually figures and the legend and the related text is in one place, right?
I mean, related text is still somewhat here and there, but at least the figures and the legends are closer.
This again is limited by the fact that we prepare our formats with the PDF in mind, right? Because the PDF, you have to get 4 big figures, and that one figure has to contain some 8 panels because there's a lot of data. This is all limitations of printed format. If you were just doing this for the web, there's no reason you need to have only 4 figures, right? You could have 20 figures. It doesn't matter. You just bring a figure when you're talking about the result in your text.
Yeah.
But we're stuck in this loop.
That's true, dude. Yeah.
Of publishing, and there's so many other things, right? So initially, when this was print only, page printing costs are expensive, which is also why everything is like squeezed together.
Yeah.
And that means now a normal page, instead of having text all across the page, it's like shrunk to one corner, and you have like these multi-column layouts. All of this is great if you want to save cost when printing paper,
right?
But not good for reading.
Yeah. Yeah, actually this also reminds me of this episode I was listening on the Night Science podcast, where this scientist, I don't remember her name, she was also talking about how basically the evolution of publishing research. Initially, obviously, there were constraints because exactly of the printing thingy. You have to have only these many words, only these many pages and stuff like that. And we still have that, and then we have not changed it at all.
Yeah.
And that limits the way you convey your results.
Exactly.
Like not just visually, but also like comprehending it, the way you're putting it out. You can now actually say other things. You can, also talk about how the discovery was made and stuff like that. It can be changed, but it is still the exact same. We did this and then this was done, and then we observed this, the same words. So I don't know how or when that would change. Hopefully soon.
Yeah, well, at least on the formatting side of things, I would say things are changing, right? Like I said, most modern journals, the web interface is quite usable, right? It's everything is like hyperlinked on the left. You can jump between sections easily, and if you're on the computer automatically, that means you can control F stuff, and that's way easier already than reading physically printed out papers. Which, if you are doing, please stop. I beg you, for the sake of humanity. But the thing is, the problem is not solved though. Like for example, a lot of other journals, their websites absolute trash. It's built in the 1990s or something, and worse than the PDF somehow. I don't know how they managed to do it. Seriously. So that's where like a few tools like Readwise Reader come in. I like them.
Actually, I've never tried any of this. Maybe I should give it a shot.
Yeah, I mean there are other more conventional tools that have been doing it for longer, like Pocket and Instapaper. So these are basically read it later apps. And the main purpose for them is like if you come across something on the internet, you just hit save and you can come back to them and read it later. But the difference is that they have sort of taken all the text in your article and parsed that nicely in like a easy to read.
Is this like a web plugin? Or?
It's a web plugin, it's an app, wherever you're reading. You can so for example, you can just press share and send it to your app and it gets saved. And you can read that anywhere from any device.
That's pretty cool.
Yeah, and usually they'll have more features like highlighting, you can take notes within the app, all that. But I like Readwise Reader specifically because it works for paywalled links. So normally Instapaper and all, let's say this article has a paywall, but you have access to it. When you share it, it no longer works. It's behind a paywall. But this app does work.
I see. That's how you're sending it. I see.
Yeah, and the nice thing about this is now I have something paywalled, I have access through whatever access I have, I can also share it with you.
Yes.
You don't have to pay again.
Yes.
Which is
That's
cool.
awesome. Yeah.
Yeah.
Very cool. Some of my other posts are a bit more scientific. I don't know if I want to go into it.
What have we done so far? Let's
Yeah.
see if we've covered all our types of posts. This was like random finding, dumb finding even. This was also same genre. You did like a question. This is like an opinion. This is also kind of a question. I think we have space for some biology, right?
Okay, let's do it then. Which one shall I do? I have some. Maybe I will just do the Xist RNA one. Yeah, so maybe I will just describe. So I also write things about my own research and just things parallel to it. Whatever I read and I find interesting, I'm like, okay, let's just share it into like a blog, like a paper, journal club thing, or just something that I read by myself. So one of the papers that I read was about these Xist RNAs. So females have 2 X chromosomes and males have 1 X and 1 Y. So to balance that in females, early on, one of the X's is kind of shut down. And how that happens is there is this RNA called Xist RNA that goes and binds to the same X chromosome and tries to inactivate it by some signals that shut it down. So in the field, for the longest time, it has been known that it only shuts down this X chromosome and not the other chromosomes which are there in the cell. But this recent paper found out that actually this Xist RNA can bind to some other autosomal chromosomes as well. So genes which are not necessarily part of the X chromosome. And that can obviously have like a bunch of effects. So so they basically, this is I think only sometime this year. But yeah, I mean, I was just thinking about how that could actually affect a lot of developmental things in females if you have such a major change so early on during development.
Yeah.
Yeah.
Yeah, I mean, I haven't looked at the paper, but it does raise a lot of questions. Like right now when you were explaining, I was just thinking, how does only one X chromosome get inactivated, right? Like what is differentiating between that and the other
Chromosome
one?
1 and 2, yeah. I'm sure there is research on that as well, which one gets this on to start with. It's pretty cool, yeah.
And what about, like if one of the X chromosome is missing, does the Y chromosome do nothing for females? Like the lack of the Y chromosome does not need to be compensated, is it?
I'm not sure if the Y chromosomes has other genes
Apart
other than
from-
like reproductive male genes.
Yeah, possible.
Yeah, or maybe they do, and then that's why we there are like some disparities. Who knows? Yeah, but this one was quite scary to me.
Because it's binding elsewhere.
Yeah, dude.
I don't know. I feel
If
like
it
I
is-
would not take these I don't know. I have to look at the paper, right? But
Yeah, I mean, of course, I did not read the entire thing in a lot
No,
of
but-
detail about which genes are exactly being affected.
yeah.
And eventually, I think it says like after some point in development, that shutdown also gets released, and these genes again do express. But if
Yeah.
what happens during those few weeks of
Yeah.
development?
Yeah. No, but the thing is like this is not something you can control, right? It's not like you started eating something new and then
Yeah.
the people are like, oh, this new thing could do something. This is
Yeah.
literally just development.
Yeah,
How
yeah.
can you not?
I mean, obviously, I can't do anything about
Yeah.
it, but it just now posits all these things about, man, what could this do?
What could this do? But the thing is, women have been around for millions of years. I don't know if it's a real threat. That's what I'm saying.
Right, fair enough. Yeah, I mean, I was also then thinking this paper came up because of another post that I wrote, which was about the developmental mosaicism. So, in my research overall, just looks at these cases of lung cancer in non-smokers, who are also mostly females in the East Asian population. And this paper was trying to see that a lot of patients have multiple primary lung cancer tumors in the same patient. So normally you would have like one tumor or like, and then eventually it would either metastasize or however the treatment is happening, depending on that, things will change. But in these 10 patients or so, they found multiple primary tumors, which could either be because this person had a germline mutation, which means a mutation which happened, which was passed on from their parent in their egg, from the egg or the sperm, and that's why they have it in all the cells. That's why it's called germline, and then that increases the chances of having more mutations, and then just increases your chances of having cancer.
Yeah.
That's one hypothesis, and another one is just that, there was this one tumor which then spread into multiple metastasized, and that's why you see several tumors in this one lung. But what they found was, in several of the patients, it wasn't germline and it wasn't even metastasis from whatever analysis they did. And then they thought whether this could be because of developmental mosaicism. What that means is you had a normal zygote, but as you were going on during development, at some point a mutation happened in one cell. And then now, as you're growing into an embryo, this mutation is going to be peppered into your organs in different regions, locations. And now that's why you have, like, within the same lung, there are spots where you have this mutation, and then that can just increase the chance of having those tumors.
Yeah.
And that is why then I thought maybe in females, because of this XN activation thingy, does it change things? And that is why it makes females more prone to some such events. Like now, even this question of why these non-smoking females have a higher chance of having EGFR mutation. Does it have to do anything with this initial developmental Xist RNA nonsense?
I see.
How can it impact developmental mosaicism? How can it impact particular genes which have eventual roles in development, disease, and stuff? Yeah. So that was my rabbit hole. And that's why those two papers together. Yeah.
Nice. Yeah, this one was interesting. I liked that. I don't know, again, did not read the paper.
Yeah.
But based on your explanation, I liked the conceptualization, I suppose.
Yeah. It was a cool paper. We did this for Journal Club.
Yeah. But what kind of surprised me, I think I made a comment here. Something like 4 out of the 10 people had mosaic mutations, right? So that just had me thinking how frequent developmental mosaicism is in like normal humans.
Yeah.
This showed up because I guess EGFR, cancer.
Because cancer came up and then they went ahead, but otherwise you wouldn't know the frequency of something like this happening. Because why would you?
Yeah. So I don't know, because what the conventional biology wisdom is that like the cells in your body are mostly identical, right? Genes wise.
But this also why would be this so rare to happen, right? It's just a matter of one mutation in one cell and then this could happen. But we would just not know the frequency of this because why would you sample so many normal tissues?
Yeah, but then this means that then actually you are not identical, right? Your cells are not actually identical. If you had some early event that is actually quite spread through your body.
Yeah.
Which could be interesting. Like what if, I don't know, you give, for example, I don't know, all of these 23andMe, right? I don't know what sample they take. But that comes from, I don't know, the part that's not representative of your actual genetic information.
Fair enough. I don't know actually what they take either.
Some
blood?
Because blood would be hard to give. I would think they'll give like saliva or cheek swab or something. I don't know. Because it's just some normal commercial company, right?
True.
No, I was thinking, my initial thought was this must be rare because of how it's taught to us. But I don't know the actual numbers.
Yeah. Same vibes. Like we were also just thinking, damn, like how frequent is this now? It only came up because you saw cancer and multiple presentation and stuff like that. But, well, if you're not having any other consequences, then maybe it doesn't matter.
Yeah, I mean, it doesn't matter to you, but I guess scientifically that's a bit of a different understanding, right?
Yeah.
Like, then again, if you're, let's say, assuming this was very heterogeneous. So then when you're doing variant calling in tumors, then perhaps just taking one blood sample is not enough.
Yeah.
Because that's not representative of the true germline status, right?
Yeah.
Who knows?
Yeah, true. How are we doing time-wise?
Should be okay, I guess.
Yeah.
We can wrap it up.
Cool. What are your plans with this subreddit eventually?
Yeah, I mean, so right now, well, it started off private.
Yeah.
But now it's like semi-private.
What does that mean?
Which means that we have to approve people to enter the community.
Yeah.
And only those who are in the community can post. But if you share it, other people can see the posts.
Acha.
Yeah. I don't know if now somebody who's not in the community can even find it. I guess they can, but I guess who's searching, right?
No, I think you can't because I remember...
No, in the beginning it was private. But then halfway through I changed it.
I see.
Changed it to half private. The reason was obviously first, I mean, very unlikely, but I was like, we didn't want some strangers.
Haters.
Some haters. But more so, I guess, to feel like that's a safe space to post stuff, right?
No, I think that it's a nice soft start and then you sort of test the waters, get used to it and then expose
like
more.
some of the people who helped me start, one person, I can't say the name because I was her concern, I guess. She did not want to be known by her real name. So hence we chose Reddit, where you can have anonymous usernames. But beyond that, also some of them are sort of, we are talking about things we like fucked up, made some mistakes, and I guess not everybody is as comfortable sharing that, especially to strangers. So right now it's in this like semi-private zone, but and maybe it will stay that way. I'm actually okay with that.
Yeah.
But definitely looking to get more people involved, right? Have more people posting. Start with friends, I guess. Right now we're still in a narrow number.
Yeah.
But eventually.
Yeah. This is very fresh right now, right?
Yeah,
Like only a few weeks. Yeah. I think it's doing well.
Any thoughts?
I think this has become like a great platform for me in general, because I told you a while ago I wanted to try some writing, general writing, which I do like just more casually, but now this is just like a more proper platform to just do it and like put it out. It just has more value. There's other people who can read it, have something out of it, have some discussions. Also, I think it also helps me understand my own things better, because obviously writing just improves your understanding of things. Definitely. I think several things which I was reading anyways, like this paper, blah blah blah, but now if I want to write it in a way that, even if it is obviously not super detailed and not filled with information, but the take home message has to be there. So I need to understand the take home message to start with. So I think that's really, really helpful, and I enjoy that.
Nice.
Yeah.
Yeah, I've also been, there were a few things which I did, like you said, sort of read through to completion, only because I wanted to post it.
Yeah.
Otherwise I would have stopped halfway and be like,
I get it. Exactly. And that happens to me all the time.
I see.
And just like even papers which are just useful for your research or something you found exciting, interesting, it's just nice to do it more formally. Yeah. It's good practice. It's great practice.
Cool.
Yeah. I would want to do more. Yeah, I think as I mentioned, I work in sports, so I just.
So you had a big spurt.
Yeah, I did.
You vomited a few
I did.
And then you stopped. Okay.
But I still have like some things here and there, which I think it's just a matter of time.
So you're collecting again till you get a big batch.
And then I sit and then I just like get on with it.
Sounds good.
Bit of I think momentum just like gets on, and then it's positive feedback loop, and then like go. It's nice, yeah.
Cool. So I guess if any of our listeners want to join, drop me a message and I will do a little interview and then let you in.
Anything to wrap up?
Closing thoughts. Last words.
Damn.
I don't know. Something different?
Nothing, sub bro?
I mean, we chatted enough.
We did. I guess let's just end it then. Okay, cool.
Okay, cool.
Bye.
Bye-bye.